In the last video, we lumped bioenergetics and metabolism together. Here we're going to separate them just a little bit to kind of hone in on true metabolism. So metabolism includes bioenergetics, or taking of foodstuff and turning it into energy, but it also includes a multiple of other things, and this includes the sum of all chemical reactions that occur in the body. So bioenergetics is metabolism, but metabolism isn't only bioenergetics. It contains all the reactions, and those number of reactions are a lot. And a lot of the semester will actually be talking about these reactions in this next several modules. We're going to talk about them as they relate to bioenergetics specifically, but we'll get into them in other ways, such as how we build muscle, how cardiovascular system changes, protein-to-protein cell signaling, et cetera, et cetera. So before we get in too deep, let's continue the intro theme of this module and talk about the two types of reactions that can occur. The first is anabolic reactions. Anabolic reactions are taking simpler substances, combining them together to make more complex molecules. In my head, the way I think about this is think about anabolic steroids. We use anabolic steroids to help grow and build muscle, make it bigger, and that's the idea of what these reactions are taking place. Is we're taking simpler, smaller parts, making them bigger, more complex substances. The opposite of this then is a catabolic reaction. This is breaking down complex molecules into smaller, simpler structures. So for example, at least when I think of muscle atrophy, that's a catabolic process and that we're breaking muscle down. Or we can think about this simply as taking glucose, breaking it down into single carbons to pass through the electron transport chain. For any and all of these reactions takes place, there is energy being either used or produced in both of these situations, right? And the energy is coming from the chemical bonds that exist between atoms, and they contain a relatively large amount of potential energy. So in bioenergetics, we need to take the energy stored in the bonds of food, stored in those carbon bonds, and convert them to ATP. There's two different reactions that will take place. Some of them will require energy to be added to before this will take place, and that is what we call an endergonic reaction. So first we'll say that, again, as I mentioned, all chemical reactions involve a change in energy in some form or fashion. Right? So endergonic reactions, these are in which the energy must be added. So energy is required before this reaction will proceed. We call this a positive delta or change in energy. An example of this is photosynthesis, right? So we take reactants, we put in energy, in the case of photosynthesis, the energy comes from sunlight, and we make products which in general would be some type of carbohydrate produced by the plant. Right? The opposite of this then is an exergonic reaction. Exergonic reaction causes energy to be released, often in the form of heat. These have what we consider a negative delta E, or negative change in energy. And again, an example of this is glucose breakdown, right? So we take glucose and its molecule, kind of strip out the energy found in the bonds between glucose, and use that energy and harness it into making ATP to produce energy in that way. It is very common to use energy released from an exergonic reaction to then push forward an endergonic reaction, right? So we couple this. So this one needs energy, this one releases energy. So if we take this energy and put it into this equation, we can push that reaction forward. This process is called a coupled reaction, whereas we take the energy-releasing exergonic reactions here in this model that we're using gears. So this is producing energy, which is then used to spin the endergonic reactions. Again, this is what we term endergonic and exergonic coupled reactions in order to kind of push these through. A very common coupled reaction is an oxidation reduction reaction. So let's take a look at what an oxidation reduction looks like. So anytime there is a loss of electrons we consider this an oxidation. So here, this electron is lost from molecule A to molecule B. That loss of electron is considered an oxidation reaction. You may have remembered this from your science class, from chemistry, or from biology. Let me see if I can grab a pen real fast. And so we look at this as oil, and then we'll get into Rig, but oil stands for oxidation is loss of electron. That is, in general, coupled to a reduction, which can be, goes for an oil rig. Sorry for my poor handwriting. So Rig here stands for reduction is gaining. So what we see is the loss of an electron from A to B. A is oxidized. Again, oxidized is lost, whereas B is reduced. Reduced means gaining an electron. And again, these are coupled reactions because a molecule cannot become oxidized unless it donates electrons to another molecule. So you have to have both of those. I'll make this comment. So in a lot of the reactions that we're going to be studying, you actually won't see, in this case, we have our nice big E here. So you won't really see very often that an electron is actually lost. Indeed, what we usually see in these reactions is the use of a hydrogen atom being lost or gained in the reaction. That's because a hydrogen ion only contains one electron. So it is essentially the same thing as passing on a single electron, but in almost all biological processes, we'll see that this is a hydrogen ion as opposed to an electron itself. So let's take a look at what that looks like. So the two common forms that we're going to see in bioenergetics of metabolism of these coupled reactions are NAD, which stands for nicotinamide adenine dinucleotide, and FAD. These molecules are in their oxidized form. When we reduce them by adding a hydrogen ion, we now get our reduced forms of NADH and FADH, which actually accepts two hydrogen ions, so FADH2. So how do these work in metabolism? So first is we have an oxidation reaction. Again, as we talked about, oxidation, oxidation is the loss. So here we have a loss of hydrogen ions from our molecule that is being accepted. So NAD here is being reduced. So here is our reduced form. Again, I like to think of oil rig. And in this situation, it is somewhat confusing. I don't love the way that that this is expressed. It's counterintuitive, but anytime you see this NADH here, the one that is accepting the electrons and becoming reduced, is what's considered the oxidizing reagent. So if it's reduced, it's an oxidizing reagent. Confusing, I know, you'll just have to remember that. So then if we look at the opposite of this reaction, a reduction reaction, which include adding electrons to a molecule here. So here, this one is being reduced. We are gaining our hydrogen ions, and here is our oxidized. Again, the counterintuitive nature, our reducing agent donates electrons, so that's what we're doing here. Electrons, again, in the form of a hydrogen ion, not in the form of a true E or electron. We lose this, so therefore NADH is the reducing agent despite being oxidized, because what we're really looking at is the molecule that it's having the effect. This is being reduced by adding an electron, and therefore NADH is the reducing agent. So one thing I want to make clear as well, so that may be confusing, is as you hear oxidizing agent, you can look on this figure and oxygen is nowhere to be found. So just remember, oxidizing doesn't actually refer to oxygen whatsoever. Okay. The next thing that we're going to cover in this section is how these reactions actually proceed when we need energy. So, for example, if we need a lot of energy in order to get a reaction to proceed, and that energy isn't available, or it's just some astronomically high value, how do we do that? We do that through enzymes, which will be the next topic that we'll cover once I clear all my drawings off here. Enzymes are used in the body to help regulate the speed of which a chemical reaction takes place. I like this figure here. This is from a different book, but the idea is that there is some type of activation energy that must be included into the reaction before the reaction will occur. So you can see the activation energy here is shown by this large purple square. What enzymes do is they ultimately reduce the activation energy, so you can see that the purple square goes down, allowing the reaction to proceed at a much faster rate. So therefore we call enzymes protein catalysts, or else they help speed up the rate of chemical reaction, again, by lowering the activation energy. So how do these work? Primarily they work by putting the substrates or the products or reactions that we're trying to produce in a very close proximity to allow that activation energy to be loaded. One of the most common models, and the one that I'll present to you here, is the lock and key model. The lock and key suggests that there is a very specific key, and this is true of enzymes. Enzymes only take a very specific substrate or reactant into its complex, and so if that key in theory doesn't fit, then that enzyme won't be able to use that substrate. So in this case, substrate, we're using an example of sucrose here as our substrate. Sucrose is a reactant molecule, comes into the active site of the enzyme. Again, this active site specific only for sucrose in this situation. The substrate fits in, forming an enzyme substrate complex. This ultimately lowers the activation energy, which allows the bond to be broken of sucrose and be then converted into glucose and fructose, which then exit the enzyme. So one thing you'll note about the enzyme is that it is not consumed or produced in this reaction. It's only there to help facilitate the speed of the reaction. We don't use the enzyme, and in theory this enzyme can now come back and perform this reaction again and again and again. Okay. So enzymes, again, cannot change the total amount of energy released by a reaction. All they do is change the activation energy. They don't change the amount of energy released from a reaction. And then how can we recognize what enzymes are? Almost all enzyme names end in an ACE, as in ASE. So for example, a kinase. A kinase is known for adding a phosphate group. Dehydrogenase, or removal of hydrogen enzyme. Oxidase catalyzes oxidation reduction reactions. Isomerases, rearrangement of the structure of a molecule. So again, all four examples all end in ASE, which is very common nomenclature in enzymes. So how can we modulate enzyme reactions? So one of the ways that we can modulate them is what's called the mass action effect. Your book does a good job talking about this. This is the idea that the concentration of substrates and products will ultimately determine which direction a reaction goes. So if you have a bunch of product in one side, of course it's going to go then to the opposite and vice versa. So example, given that A and B combine to make AB, a higher constant of substrate A and B will cause the anabolic or the combining production of AB, whereas if you have a large concentration of AB, then that will push the catabolic reaction of B and A. The other two important modulator of enzyme activities are temperature and pH. Both of these are very sensitive to both a increase or decrease in temperature. In general, an increase in temperature will cause the active site of the enzyme to kind of denature, unfold, and not work properly, whereas a change in pH does the same. So indeed, this is very important in exercise physiology because as we exercise, in general, the body temperature or temperature of the muscle increases, which can ultimately affect enzyme kinetics, as well as pH goes up from the production of lactic acid. So they both change. So indeed, we have some enzymes which become less functional, and we even have some pretty cool enzymes that become more functional with an increased temperature or a decrease in pH with acidity, and we'll talk about those as the course progresses. But for that, that's going to end our talk on bioenergetics introduction. In the next module, we'll get into ATP production from the creatine phosphate system and glycolysis.